Hello, everyone. Welcome to Global IMMUNOTALKs. It's a pleasure to be here today with you. I'm Marcelo, and I'm going to host today Marcelo TorresBozza. And hello, welcome.
Hi, Asia.
So today we are going, as you know the drill, we are going to ask few questions, describe a little bit of your career. There it's going to be your presentation. And in the end, as you know, we don't have the questions directly at the talk. You're going to post them and you're going to have the slide introducing how you can communicate. And Marcelo is going to answer your questions online, and it's open for a while. So thank you again for joining us today. And maybe I will go briefly a little bit to tell about your story, and then I will ask you a question, a surprise question. So Marcelo received this, actually he's a clinician, medical doctor from the State University of Rio de Janeiro. And after doing a postdoctoral fellowship at Harvard, he continued to start his lab at the University Federal de Rio de Janeiro in Brazil. And since then, he also had kind of some slips to other places. For example, he had some time in the Sorbonne University and even in the Museum Histoire Nationale in Paris. He also spent some time as a Fulbright scholar at New York University with Dan Littman. And he's doing some really interesting work over the last 20 years, trying to understand the selective molecular and cellular aspects of immune inflammatory responses, which cause damage to the organism either by the direct infection agent, or actually by the host response to the infection, which is a really interesting field in my opinion. And his research demonstrated the critical role of the cytokine MIF in the inflammatory responses, in tissue damage, in mouse model of infection. His group was the first to demonstrate that heme induces inflammation through the activation of the immune response, immune receptors, and playing a critical role in the pathogenesis of the disease, of hemolytic diseases. And his recent work on reactive oxygen species benefits all the intracellular, how they benefit the intracellular parasite while antioxidants are reduced both the parasite load and the tissue damage in different mice models. And today we are going to hear his talk about disease tolerance influences the composition of the microbiota and the susceptibility to inflammatory bowel disease. So it's a pleasure to have you here today. And I would like to ask you, how do you handle setbacks and unexpected results in your research?
Asia, thank you very much for your introduction. So I would divide this question in two. So the first one, how I handle setbacks. I will consider setbacks and the unexpected results as two different things. So the setbacks, I would say, in general, we would consider setbacks things that happens once in a while. So in the way I see, it's quite different. So the setbacks here, in the conditions we have doing research, it's something expected. So we do have a continuous of setbacks. And by living in these conditions, I think that we gotta be resilient and to have a lot of enthusiastic momentum to keep the group motivated and able to do the research. And connected to that, I would go to the second part of the question, is how I handle unexpected results. That I think is a very important issue, because in general, and probably the most interesting findings we had came from unexpected results. And I remember a while ago, I was watching an interview from Anish Kapoor, that is a contemporary artist from India and Britain. And he was asked about his work and like how he would talk about his work. And he was saying that he was much more interested in the experience of doing the work than the final result, because during the experience of doing the work, he would consider that like experiments. And when these experiments fail what he was planning to do or to obtain, it was exactly the moment that he would find avenues to new works and to new ways of seeing things. And what I will present today is very much in this direction. So it was based in a quite unexpected result, and how we handle, I think, is with a lot of enthusiasm, I would say.
It's kind of when a prepared mind meets serendipity. So it's all about being there. So thank you very much. And without further ado, please, if you can share your work, your slides, and again, you can also follow us. Again, as you know, these talks are regular, and you can then see them online in YouTube. Please.
Okay, Asia, thank you a lot. So I will present this story, and I consider that, as I mentioned, it's a quite unexpected result, and it completely changed the direction of the research, and that's the story I will present to you today. But the general idea is related to how hosts and microbes interact, and we have several different ways, but we could group as these main relationships as commensal microbiota, pathobionts, pathogens, and no infection at all. So some hosts from a given species would be prone to have certain commensals and certain pathobionts and pathogens, and or be unable to be infected by a certain microorganism. And of course, this is related to the species, but is also an individual issue by itself. So meaning that for certain individuals, a pathobiont could be a pathogen, or a pathogen could be part of the microbiota. And this is a long process of coevolution, and this coevolution accommodates the possibility to deal with these microbiota, and at the same time to be able to retain the ability to eliminate pathogens. So the way we do, the hosts will do that from plants to animals, is basically based in two main strategies. So these two defensive strategies are resistance, one of them, that limits the negative impact of infection. And basically what resistance will do is related to reduction of the microbial burden or the microbial load. So the idea would be how we fight infection. And of course, the main system associated with this function is the immune system. It's not the only one. So of course, we have nutritional issues as well that can control a microbial burden. But the immune system is the main system associated with the resistance. So as you can see here in this graph, so fitness would be a health state, and as the pathogen burden would increase, the health state would go down. So in this ideal scenario, resistance, if achieved, would control the pathogen burden and would bring back the health state. So that would be an ideal scenario, but in fact, that's not true, as I will show to you. So we have a second way to deal with infection, and this second way, it's called disease tolerance. And the general idea of disease tolerance compared to resistance is that the disease tolerance mechanisms are not associated with direct control in terms of pathogen burden, but in fact will impact how we deal with the infection in terms of our cellular and our tissue damage response. So it would help to control the cellular malfunction and the tissue damage associated with the presence of the infection and with the presence of the immune and inflammatory response. And as you can see in this figure now, let's say a 10 to the 7 arbitrary unity, we can see that the green line, it's someone that is still healthy or with a really mild disease and can go to work and exercise and have sexual relationships, while the one in orange, it's severely with symptoms and signs and symptoms of disease, and the one in red is eventually almost dead. So basically what this graph is indicating is that with the same pathogen burden, the individuals might have a totally different outcome in terms of health and disease. And these concepts and studies have been championed by David Schneider, Janelle Iris, Ruslan Medetov, Miguel Suarez, and many others. But the point is that one would need to see that to every infection response, we have both the resistance and the disease tolerance coordinated, working in terms of defense. And this could be represented in these ways. So we can imagine that to a certain infection, someone would be like high tolerant, and this high tolerance means that the impact in cell malfunction and tissue malfunction would be very low. So the high tolerance means that the individual can deal with the infection and with the response. So the low tolerance would be the opposite. And the high resistance would be someone with the ability to clear the pathogen, and the one with the low resistance would be the one unable or less able to deal with the pathogen control, and that would be, let's say, someone with immune deficiency. Of course, that this is something graded. So as you can see in this figure, the idea is that you actually would have the possibility to be in one of these quarters. And the idea is that always to every infection, we will have both tolerance and resistance playing a role. And one very interesting example that probably was the first example that shows how resistance and defense works together was the case of Mary Mallon, this Irish cooker that came in the end of the 19th century to New York to cook in a wealthy family. And she eventually was found to be the source of the outbreak of a Salmonella typhimurium. And actually, she was basically healthy and was considered responsible for spreading the outbreak for around 3,000 people. And again, I mean, the point is that if we think about the idea of resistance and tolerance, so Mary Mallon would have like low resistance to the bacteria, but she had like high tolerance. So she was barely symptomatic. So she at that time, in the beginning of the 20th century, there was no vaccine or no treatment, and she was put in quarantine for life. So the inflammatory bowel disease is one of these conditions that we would consider both resistance and tolerance when we think about the disease. But it's interesting to observe that after the Second World War II, especially after the 60s, we see a huge increase in the cases of Crohn and ulcerative colitis. So this is an inflammatory disease that is associated with the presence of the intestinal microbiota that eventually could contribute to the inflammatory damage. So it is now well appreciated that the gut is such an incredible environment in which we have in homeostasis the ability to deal with this huge amount of bacteria, viruses, fungi, protozoas, and helminths. And these in homeostatic, and of course the diet and all the antigens and so on. So the ability to deal with that requires the anatomical characteristics of these intestine and the innate and the adaptive immune system well balanced, and the presence of course, the mucus and so on. So we had a major change in terms of diet, use of antibiotics, and the prevalence of infectious disease over the last 70, 80 years. And as one can appreciate in the second panel, we have a change in terms of the microbiota, what we would call dysbiosis. And dysbiosis is associated eventually with a damage to the epithelial barrier and entrance of these pathobionts in the gut and causing in the tissue gut, causing inflammatory response. And of course, this is associated both to, as we discussed, with the host genetics, but also with the changes in the environment. And as you can see here in this figure where it says genetics, we might have changes such as defects on IL-10 or IL-10 receptor that would impact on a tolerance, or changes in receptors such as NOD2 that would impair resistance. So it's a quite interesting scenario to think about resistance and tolerance. And this was actually demonstrated a while ago when Kuhn and Kohler showed in '93 that IL-10 deficient mice would develop an inflammatory inflammation, an inflammatory disease in the gut. And this colitis was actually associated with the presence of the microbiota. So in the conventional husbandry, so the animals would develop a fast inflammatory bowel disease, while in the SPF it would take longer. And in germ-free conditions, the IL-10 deficient mice would not show any inflammation. So this is a clear case in which the animals present low tolerance to the presence of something that it would not be pathogenic, that is our microbiota. And it's quite clear that IL-10 is a master regulator of inflammation, because in several sterile and infectious models of inflammation, lack of IL-10 has a major impact. As you can see here in the survival curve, the IL-10 deficient mice, when infected with Toxoplasma gondii, they die very quick, while the wild type and heterozygous are fine. But the point that I want to make here, it's clear that based in the SAJ2 gene, that the IL-10 deficient mice is able to clear the parasite at least similarly or even better than the wild type. So that means they have slightly higher resistance, but by being low tolerant to the presence of Toxoplasma gondii in the gut, they become very sick and die in 2 weeks. So there are several examples, such as in sepsis, endotoxemia, Chagas, leishmaniasis. So there are a huge number of examples in which we observe that IL-10 or IL-10 receptor deficient mice, Helicobacter hepaticus, the work by Dunn. So there are several examples in which the deficiency of IL-10 has a major impact, mainly in the ability to deal with tissue damage. So on the other hand, there is this cytokine, the macrophage migration inhibitory factor, that was described in '66 by John David, and later the work from Rick Bucala really moved the field. But basically, MIF is a pro-inflammatory cytokine that controls the production of several inflammatory mediators, but also it's associated with a suppression of p53-dependent apoptosis. And overall, both in the sterile and on infectious conditions, MIF seems to play a major role. So the absence of MIF makes animals less prone to inflammation and tissue damage, both in allergic and autoimmune, as well as infectious disease. And our group has been showing that in different contexts, including viral, bacterial, protozoa, and helminth infections. So in these different cases, what we observed is that the lack of MIF is associated with or reduced or similar resistance, meaning a pathogen load, but a better outcome in terms of survival and tissue damage. So in this case, it would be that the MIF deficient mice are more tolerant. And it's interesting to observe that it's not really related to TH1 or TH2 or TH17. It seems to be more general, similar to what happens with the IL-10 deficient mice. As in opposition to IL-10, as you can see here, in both sterile models of a IBD, one is the transfer of CD4 positive, CD45 RB high cells. What we observe is that to HAG deficient mice, so the HAG MIF deficient mice present less inflammation compared to the transfer just to the RAG mice. And the same is observed in the DSS model. So again, in opposition to what's happened to IL-10, so the MIF deficient mice are less susceptible to the gut inflammation. And again, in opposition to what's happened in the IL-10 deficient mice infected with Toxoplasma gondii through oral infection, what we observe is that the MIF deficient mice survive better to oral infection. They have less histopathological scores, and this despite the presence of more Toxoplasma in the gut. So this would be exactly the opposite. So they would be low resistant and high tolerant. So all these issues made us to come to a very simple idea. And at that time, I received a student that was not very used to work with animals and bench experiments. So I said to her, so let's cross the MIF and the IL-10 with the hypothesis or with the idea that the double knockout would somehow be protected from the different models of infectious disease. So the question would be, what phenotype is dominant to be high tolerant or to be low tolerant. So what would be more important? So the question could also be, is IL-10 important to the high tolerance that we observe in the MIF deficient mice, or is MIF contributing to the damage that we observe in the IL-10 deficient mice? So that was the very simple proposition and the very simple experiments. But after 10 days, the student came to me and said, Marcelo, I put the animals to breed, and something very weird happened. So all the IL-10 deficient mice die when they stay together with the MIF deficient mice to do the crossing. Then I said to her, okay, so why don't we do a real experiment out of it? So you go back, you put MIF and IL-10 deficient mice, or wild type together with IL-10 deficient mice, or just IL-10 deficient mice, and you go and you record the weight and whether or not they actually die. So that's what she did, as you can see here. So the animals, all we see here in these curves are IL-10 deficient animals. So IL-10 deficient animals that were kept just with IL-10 deficient animals, IL-10 deficient animals that were co-housed with MIF deficient mice, or IL-10 that were co-housed with wild type. And as you can see, the ones that were co-housed with MIF lose weight and they die in 2 weeks. So this is very robust and reproducible. As it's shown here, the IL-10 that were co-housed with MIF deficient mice present a rectal prolapse. They present a blood in the feces, and the colonoscopy, see in the middle, we can observe bleeding and ulcer compared to the ones that are just kept with IL-10 or with the wild type. So this indicates that the co-housing of IL-10 together with MIF causes an hemorrhagic inflammatory disease in IL-10 deficient mice. Again, as you can see here, the colon length and the histopathology, only the group of IL-10 deficient mice that were co-housed with the MIF present major changes. The phenotype characterization of these cells in the lamina propria shows that the interferon gamma producing cells are increased in the gut, in the colon of the IL-10 deficient mice that were co-housed with MIF. But it's interesting to observe here that the IL-17 positive cells were increased in the IL-10 deficient mice that were co-housed with wild type. But we don't see an increase of gamma interferon in the IL-10 that were co-housed with wild type. So this suggests that the disease might be triggered by an interferon gamma producing cells, and this seems to be a CD4 positive cells. In this case, again, the presence of a gamma interferon, a CD4 positive, FOXP3 negative cells in the lamina propria. And again, as you can see, the CD4 cells from the wild type, from the IL-10 deficient mice that were co-housed with wild type, there is an increase in these cells, but this does not, it seems to be sufficient to cause disease in IL-10 deficient mice. And interesting, these FOXP3 positive CD4 cells from the IL-10 deficient mice have also an increase both in interferon gamma cells as well as in IL-17 positive cells, suggesting that there is a change in these regulatory cells that, of course, are unable to produce IL-10 because they come from the IL-10 deficient mice. And also, we observe in these animals, the IL-10 deficient mice cohoused with MIF, a significant increase in neutrophils in the intestine, in the colon. So this completely changed our original idea that was, well, okay, we're going to have these double knockouts to study in different infectious models. So at this point, it became clear that the cohousing was causing disease in the IL-10 deficient mice, so the cohousing with the MIF deficient mice. So we asked whether or not the microbiota or something else present in this cohousing that was transferred in this cohousing was causing the disease in the IL-10 deficient mice. So actually, we hypothesized that an animal that has high tolerance would harbor a microbiome that could have pathogens and pathobionts able to be transferred and cause disease in animals that are susceptible. So we did obtain just the fecal pellets from IL-10 and put in a cage of IL-10 deficient mice, fecal pellets from wild type to IL-10, fecal pellets from MIF to IL-10, and we had exactly the same result as the cohousing. So the feces is sufficient to transfer the disease to IL-10 deficient mice. So having the feces, we filter these feces, as you can see, and what we observe is that the flow through that would have virus or metabolic molecules are unable here in brown to cause the disease in IL-10 deficient mice, but the feces from MIF deficient mice, as we saw before, cause weight loss and lethality in IL-10 deficient mice. So now we know that the cause of the disease is present in the feces, and it's probably large, being or bacteria, protozoa, or fungi. So to test our hypothesis, we sequenced the 16S, and as you can see here, the beta diversity in green, blue-green, you see that the IL-10 deficient mice has a microbiota that is completely different from the wild type and from the MIF deficient mice, that they somehow overlap, but they still group separately. But when we see the alpha diversity, we actually confirm our hypothesis that MIF deficient mice have higher diversity in terms of the microbiome compared to the wild type, and even more compared to the IL-10 deficient mice. And this composition, it was interesting because, again, IL-10 deficient mice at this point was very healthy, and in fact present a microbiota that suggests that there is a healthy microbiota despite this reduced alpha diversity. In opposition to that, the MIF deficient mice present a ratio, Firmicutes bacteroidetes, that is associated with dysbiosis, and they also lack several of the groups that are associated with a healthy microbiota. So the MIF, despite being healthy, and in this case, what we are comparing are the groups without the cohousing, each one by itself. And as you can see, the microbiota of the MIF seems to be dysbiotic, despite the fact that they are completely healthy. And when we see the result of this cohousing, we observe that there is a change in the microbiota of the IL-10 deficient mice that were cohoused both with MIF as well as with the wild type. There is an increase in terms of their diversity. So we decide to ask whether the bacteria in the colon of these animals would be covered by antibodies, IgA and IgM. And as you can see here, both the MIF deficient mice and the wild type, but mostly the MIF deficient mice, has less bacteria covered either by IgM or by IgA. And as you can see, the IL-10 deficient mice had more IgM and IgA positive bacteria. And we observed the presence of IgM positive B lymphocytes, B220, and it seems it can correlate with the reduced recovery of a bacteria by IgM. In this case, the MIF deficient mice seems to have less of these cells in the colon. So now we know that the bacteria seems to be the cause, or at least could be the cause of these disease, in this transfer disease of the disease. So we decide to treat the IL-10 or the MIF or both with antibiotic cocktail, as you can see here. But somehow, to our surprise, the cocktail was unable to protect the lethality caused by the cohousing of MIF together with IL-10. So all the IL-10s still died, despite the fact that we treated them with a cocktail of antibiotics. So that, again, was an unexpected result. And we had two main hypotheses. Well, maybe it's not a bacteria, maybe it's a fungi or a protozoa, or the bacteria that are actually causing disease might be resistant to this cocktail. So that it became an opportunity, because now we had an extreme reduction in terms of the bacteria that we could recover in the feces of IL-10 deficient mice after the antibiotic treatment and after the cohousing. And we ended up with some bacteria, and we isolated them, and we were able to see that they were resistant to this cocktail, but they were susceptible to gentamicin. So that again opened another opportunity. So we decided to treat the IL-10 deficient mice and the MIF. In this case that we are showing, we treat the MIF deficient mice with the cocktail plus gentamicin, and we put the feces, the fecal pellets, to the IL-10 deficient mice. And as you can see here, the triangle group. So the feces coming from the MIF that were treated with cocktail plus gentamicin was able to protect the lethality. So among the bacteria that we isolated, Proteus mirabilis and Klebsiella oxytoca were there, and they are known to cause colitis both in humans and mice. So we decide to further analyze that. And as you can see here, especially Proteus mirabilis was very effective in causing lethality and weight loss and colitis in the IL-10 deficient mice. So an important point is that most of the studies indicate that one can transfer disease through the microbiota when the animal is actually sick, or someone with IBD might transfer to someone that live together with him or her, because the microbiota is profoundly affected by the inflammatory condition and the dysbiotic condition. But as you can see here, the column of the wild type and the MIF deficient animals are fine. So they don't show major changes in terms of histological changes, in terms of the presence of inflammatory cytokines, and they gain weight similarly. So indicating that the MIF does not seem to have a major problem in their gut, but still they transfer a pathogenic microbiome. And we started looking what could explain this increased tolerance. We don't see major difference in terms of mucus layer. We do see changes in terms of a number of mediators, inflammatory mediators, or such as IL-22 that has an important role in the homeostasis of the gut, IL-33, that's an alarmine, and also Helm-B, in this case, is reduced, and Th2 cytokines such as IL-13 and IL-5 were also reduced. So we don't know exactly what is explaining the increased tolerance of MIF deficient mice, but at this point, we were we are still going to understand what's going on and what's happening to explain this change in resistance and change in a tolerance in the gut. But by that time, we were able to do the crossings, and we obtained the double deficient mice, so MIF and IL-10 deficient mice. And as you can see here, the double knockout would not appear in a Mendelian distribution, so we obtained much less double knockouts. And if you see in the picture, the IL-10 deficient mice are much smaller, and they do present a rectal prolapse and a complete change in the gut with this major inflammation and ulcer. And in the end, that brought us the idea that the absence of IL-10 is actually completely dominant in terms of the inflammatory disease. So in the absence of IL-10, even though the animals also are lacking MIF, they still have the inflammatory disease. In opposition to that, we observed that the lack of MIF was dominant in terms of the presence of the microbiota. So the IL-10 deficient, the double deficient mice seems to have the worst part. So the microbiota of MIF and the susceptibility to damage of the IL-10 deficient mice. So these animals were, then we obtained the feces of these animals, or we did the cohouse of these double deficient mice with IL-10 deficient mice. And as you can see, they are able to transfer disease to IL-10 deficient mice. So, and we can rescue the double knockout, at least in part, using the cocktail together with gentamicin. So overall, we came with this hypothesis of the uncertainty of living together. So this means that someone that is tolerant to intestinal tissue damage might be able to have or harbor a microbial community that would have more pathobionts, and living together with someone that has less a tolerance would eventually make these individuals able to transfer this microbiome and these pathobionts, although they are healthy, and eventually causing disease to someone that has this reduced tolerance and susceptibility to tissue damage. So I would like to thank the people that actually did the work. So Vinicius Mendes is the student, the PhD student, now is a postdoc. He's an incredible scientist. and he actually did most of the experiments together in the beginning with Letícia Martimignano, Jamil Kitoko, Helena Montes Cobos, Andressa, Samara, and Guimarães and Gabriel Teixeira. So this work would not be possible without the help and collaboration of different great scientists. Fábio Canto was key, and Heitor de Souza did the histology and the colonoscopy. Batiar Yilmaz helped a lot with the microbiome analysis, and then Littman and Varsha in his lab, Hagava, help us to isolate the bacteria. So with that, I would like to thank you, and I'm open for questions.
Thank you very much. It was definitely a great example to what extent serendipity plays a role in unexpected results and what happens when you follow them. So thank you. And as you know, you can answer the questions. You can all ask questions on X, Twitter, and Marcelo will be able to answer them then. And again, you can watch the talk again on YouTube, and hope to see you next week on the next talks. So thank you very much, Marcelo.
Thank you. It was a pleasure, Asia, and thank you for hosting me today.
Bye. Have a good day, all.